validated biomarker, because that essentially is what we'll need to next do. The next paper, we have two presenters. Actually, Dr. Marileila Garcia from the University of Colorado will present, but we also have Dr. Bruce Johnson from Harvard, who's been intimately involved with the work. And the paper, abstract 1348, ALK fusion and MET amplification as molecular biomarkers and therapeutic targets in advanced lung adenocarcinomas in the Lung Cancer Mutation Consortium. Leila.
Thank you. I'm very pleased to present this on behalf of the Lung Cancer Mutation Consortium investigators. This is a study that has been funded by National Cancer Institute in the United States, and the principal investigator is Dr. Paul Bond. Dr. Johnson and I are part of the team. Next, please. So the goal of the study was to define molecular status of 10 relevant genes in 1,000 advanced lung adenocarcinomas. And these genes, these 10 genes, 8 of them were investigated by mutation analysis, and they were KRAS, EGFR, BRAF, HER2, PIK3CA, AKT1, NRAS, and MEK. And we did this using mutation analysis, and Dr. Johnson is presenting these results in probably 10 minutes or 15 minutes from now in another section. The other two markers were investigated by FISH analysis, and those are the ALK, in which we investigated the occurrence of gene fusions, and the gene called MET that we investigated for amplification using the fluorescence in situ hybridization technology. Another goal of the study was to identify the personalized therapy with either an approved agent or a trial agent that were targeted to specific mutations that we are found in these 10 markers. And the third important goal of the study was to assist in the implementation of qualified molecular diagnostic lab in academic institutions in the United States. Next, please. So we were 14 U.S. institutions, and in the map of the United States that you can see behind it is you see our logos with our location. Next, please. And what was achieved, the study started about a year and a half ago. What we achieved at that time, we developed and optimized the protocols and reagents for these 10 markers. We demonstrated that high-quality molecular diagnosis could be performed for multiple molecular markers in a period of time reasonable to selected patients for target therapy. The FISH tests by themselves take 1 week of turnaround time, and the 10 markers altogether, we could do results, we can have results in 2 weeks for all of them. This current panel of mutations in these 10 genes may be easily optimized for an increased number of driver mutations. Next, please. And looking at the MET and the ALK, that are the two markers that I'm reporting today, we investigated 643 patients for ALK, and we found 9.6% of patients that were positive. And we investigated 295 patients for MET, and 4.1% of them were positive. So as you see in this study, we are heading to 1,000, but we collected about 1,000 of specimens. We are lagging behind in the the entering data in the databases most of the time. So about 55% of adenocarcinomas that were enrolled in our studies have now a driver identified. If it's not by FISH, FISH is listed here. The other ones are by mutation that Dr. Johnson is going to cover. Next, please. So the ALK-positive patients are highly sensitive to crizotinib, and a significant fraction of ALK-positive patients were enrolled in crizotinib trials. They have excellent response. We have some that progressed. So currently, we are considering enrolling these patients in other clinical trials. The MET-amplified patients are resistant to EGFR TKIs and are sensitive to MET inhibitors. Some MET-positive patients were enrolled in the crizotinib trial, phase 1, and responded very well. And if efforts are now ongoing in enrolling these MET-positive patients in other trials with specific MET inhibitors. Next, please. So in conclusion, this study shows that ALK fusion and MET amplification are actionable molecular drivers in a significant fraction of lung adenocarcinomas, that the patients with tumors depending on these drivers have significantly better clinical outcome when treated with specific inhibitors. The FISH technology is a robust platform for testing lung cancer biomarkers with short turnaround time and low failure rate. We also show that collaborative efforts are essential to qualify laboratories for molecular diagnostic testing in a cost-effective way and an efficient way. And we are very proud to verify that the Lung Cancer Mutation Consortium study is sponsored by NCI is a very successful model for translational science, not only for lung, but for other carcinomas also. Thank you for your attention.
Thanks, Leila. I'll ask people to come to the microphone while we're doing this. I'll see if Dr. Johnson has a comment.
One of the things in the institutions that have looked at this, of the people with the driver mutations, about a third are able to go on specific trials for those agents directed against them, which is about twofold higher than what we typically see in the proportion that go on clinical trials in general at our institutions.
Well, wonderful team effort. Yes, please.
Zosha Chostek from Medscape Oncology. Could you comment on the difference between the proportion of patients you found, 9.6%, and what we've heard up to now from crizotinib presentations, which has been 4 to 5% of patients have had ALK mutations? Is a big difference?
Yeah, it's twice as much as differences. So the difference in frequency depends on the type of population that we are dealing with. This study was only for lung adenocarcinomas. These adenocarcinomas were tested in local hospitals, but they went to a central screen. So in our institution, when we talk about non-small cell lung cancer, and we tested all large cell carcinomas, squamous cell carcinomas, the frequency goes down to 4%, between 4 and 5%. Okay? For all non-small cell lung cancer. When you focus only in adenocarcinoma, there is a specific enrichment for the frequency, and that's why you can get higher frequency.
Yes, please.
I'm Naomi Rodrigue from Oncology Tribune in Hong Kong. I understand it's the same study that was reported at ASCO, I mean the same consortium by Dr. Chris. Mm-hmm. And my question is, what are you doing with the patients once you identify the mutations? You call them actionable mutations, but you don't have treatments for all of them, obviously.
So in a couple of minutes, I'll be talking about what trials we've done. Not surprisingly, with EGFR mutations and with ALK chromosomal rearrangements, there's erlotinib, but other EGFR inhibitors, and there's crizotinib for the ALK. We've been able to negotiate with the companies to do the infrequent ones, which was the goal of the study. So for instance, with BRAF mutations, which are present in 2%, we've been able to work with GSK and get their BRAF inhibitor. And for the other less common mutations, for instance, well, KRAS is a common mutation, but we've been able to work with the MEK inhibitors for a large number of the mutations. And so for those, the whole goal of this was for the frequencies of 1 or 2%. We've been able to get drugs from there and open them at the institutions that have those unusual mutations.
So you refer the patients for the trials?
Yeah, and so we identify those patients and then put them on the trials.
Bruce, outside of a trial, what do you consider should be the standard of care for a lung cancer patient now, as far as genetic testing?
Well, we have been testing patients for EGFR for 7 years, and we've been testing for KRAS for for 5 years, and for the EGFR mutants, we put them on erlotinib or a study if we have it. We put the only other one which is close to approval is crizotinib for the EML4 ALK chromosomal rearrangements, and so we put those patients on studies. The other ones, it's less available, and those studies that we negotiated for the 1 or 2% weren't. We negotiated with them starting last year, so they're just now beginning to open. We have a little bit of experience with HER2 mutants that's being presented by Leena Gandhi later this week.
Right, so it's remarkable how lung cancer is being subsetted now, and hopefully with more effective therapy. Thank you both very much. Dr. Johnson's going to need to now leave to make that presentation, but the remainder of the panel will stay for discussion at the end. Our next presenter is